Hi, welcome everybody. Today's science colloquium will begin at 3:15. I appreciate you joining us. I'm actually back at MJC, so no longer streaming from my dining room, now streaming here in my office, which is really nice. I want to tell you about a couple of things coming up. In 2 weeks, February 23rd, we have Linda Lagace. She's a photographer. She's going to give us a presentation on the birds of the Merced National Wildlife Refuge. And Linda's a self-taught photographer who's been visiting the Merced National Wildlife Refuge almost weekly during the birding season for the past 15 years. But this slide presentation will include recent photographs taken since January of 2018. Since the acquisition of her new Tamron zoom lens, Linda's been able to get up close and personal with the birds. And I just realized I'm covering my text here. The presentation will also include a brief smattering of birds Linda hasn't seen for a while, as well as some rare behavior she hopes to see again, and some application of Photoshop art filters to her photos. So I hope you'll join us for that presentation. And then we also have coming up our Modesto area partners in science is having Dirk Verdorn, Sacramento's Channel 3 meteorologist, join us. He's going to be a keynote speaker for a panel discussion that's going to be co-hosted and participated by students from MJC and Enox High School. The presentation is going to focus on the difference between weather and climate and the impacts climate can have on our environment. Dirk is an advocate for clean air and water and will emphasize the importance of being good stewards of our planet. So I hope you will join us for that. And that is back here at MJC on Modesto's East Campus Auditorium. And I believe, I don't know if we're going to be streaming that or not. You'll find out. But I know it is going to be live in the auditorium February 25th, starting at 7 p.m. I bet there will be a recording. All right. And today we have Andrew Gardner, and he is an associate professor of botany at CSU Stanislaus and also the interim chair of the Department of Biological Sciences. Today's talk is on the various contrivances by which Californian and foreign plants are pollinated by insects. It's a Darwin Day talk. I do want to mention his research interests range from systematic projects in collaboration with global partners to work on vernal pool species in the Central Valley and front yard diversity in Turlock, which sounds pretty great. I'm thinking that's plant diversity in gardens. Awesome. And I also, before I hand it over to Andrew, I just want to wish everybody a happy Darwin Day. Richard Anderson started the yearly Chuck Cake celebration. So Richard, I'll be bringing a cupcake over to you and Lynn on my way home tonight. And I hope everybody has a happy Darwin Day. And I'm going to hand it over to you, Andrew.
Thank you.
Actually, as I stop my share, there we go.
And I will begin mine.
Thank you.
All right. Here we go. People should be able to see this title slide.
Looks great.
Yeah, cool. Thank you. So Chuck has been an inspiration to me for quite a long time. I first started learning about Charles Darwin, I think, when I was in high school. And, you know, there are a lot of of great stories about Darwin. And so it's a great honor for me to be able to talk about Charles Darwin today, or any other for that matter. Um, this talk is really fun for me because I get to combine my interest in Darwin with my longtime interest in orchids. Orchids are definitely some of the plants that first really got me into botany, and so what a joy. Um, as uh, as you heard, I'm at Stan State. We're very busy, and we're working hard to expand our faculty right now. We have a lot of programs for students that are interested in coming. And so if you have any questions about Stan State or Stan State Bio or anything like that, don't hesitate to ask me either after the talk, or you could email me as well. My email is down here at the bottom, agardner1@csustan.edu. All right. So here's the cover of one of the first editions of the book. Okay, so the title of the talk is a reference to Darwin's title. And frankly, a lot of his writing was quite Victorian in its complexity. I'll start by talking a little bit about kind of Darwin's personal history, and we'll hopefully be able to reflect on some of these things throughout the talk. Charles Darwin was a pretty lucky person. He was a member of an extremely well-connected family. His grandfather Erasmus was someone who actually wrote about evolution, long before it was being kicked around in scientific circles really meaningfully. He had access to the best schools, the best mentors, any kind of resources he needed to do more or less what he wanted to. Darwin was, someone who had, kind of any number of paths he could have gone, and he experimented a lot as a young person. There was a strong push for him to go to medical school, and so he attended the best medical school in the world, probably at the time. But he wasn't really into that, and he started getting some mentors that started pulling him more and more towards biology. And what he was able to do, frankly, was to get himself on board a boat. He convinced his family to let him travel on the Beagle, this expedition around the world to explore lots of things, ostensibly to map coastlines, but they found many exploits along the way. And when he came back, he wrote about it. And the book, The Voyage of the Beagle, really kind of established him as someone who was an important voice in natural history. And he was able to kind of really establish a number of relationships and partnerships from that kind of allowed him to move in that world. Here's a picture of him at age 6. And this artist, Ellen Sharples, just to kind of encapsulate the time, often had to make paintings under her husband's name. Charles Darwin, when he started his work after he came back from his voyage, his father figured out his finances so that he could be just a gentleman naturalist. At Stan, at MJC, at so many institutions, what we're trying to do these days is to basically reduce those inequities so that more and more people have access to great schools, great mentors, great resources. So that's something that I often reflect upon when I think about Charles Darwin's personal history. Another contemporary of his that you might have heard of is Alfred Russel Wallace. Wallace was another natural historian at the time, and himself an incredible contributor across many fields of biology. And the two of them actually kind of independently arrived at this idea of evolution by natural selection. And when Wallace corresponded with Darwin, Darwin elected to work with him so that they could jointly publish their idea. And Darwin was doing this all at home from Britain, whereas Wallace was in Malaysia suffering from malaria. Wallace didn't have the personal connections that Darwin did. So after Darwin eventually published Origin of Species, which was obviously a massive thing, he was also establishing his family life and stuff. He had ample money. He had this awesome greenhouse you can see here on the upper right of this screen. And he was looking for all kinds of other ways to investigate his ideas about natural selection. And one of those sort of arenas for this work was in the area of orchid pollination. Darwin had read this book by a German botanist named Sprengel that was kind of one of the first books that looked at orchid pollination mechanisms. If you've ever looked carefully at an orchid flower, you'll see that they're really different from a lot of other flowers, and they have these really elaborate structures within them. Darwin was aware of this book, which had kind of been ignored, but he was really intrigued by it. The other thing that Darwin knew was that many prominent naturalists of the time believed that orchids were mostly self-pollinators. There wasn't much evidence of insects visiting flowers, and people had even argued that insects were maybe thieves. They were just stealing pollen or nectar, but not actually effectively pollinating them. Darwin disagreed. He wasn't convinced of this, and he started to acquire more and more orchids, both from his own activities in England, but also by corresponding with new friends and professional colleagues who would send him plants from all over the world, frankly. So he started growing them and investigating these pollination mechanisms. So a couple of years after he published Origin of Species, he published this book on the various contrivances by which British and foreign orchids are fertilized by insects and the good effects of intercross. Here's the title of the book. And these are a couple of the orchids that Darwin talked about in here. These are orchids native to Britain. So here's a little animated GIF of Ophrys epifera, the bee orchid. And what you can see hanging in the middle is a polinium, and there's kind of activity, and then all of a sudden it's stuck to the stigmatic surface of the flower. It looks like, at least. And so Darwin was wondering, if we think that all of these weird structures, all of this is totally purposeless, like, why would that be? Why would it evolve this elaborate morphology if it's simply self-fertilized? So he put together this book that was kind of a collection of all these investigations that he'd done through the years. And he wrote it again in kind of a semi-popular style as the book, his Voyage on the Beagle book was written. And it actually sold surprisingly well, a small book about orchid pollination mechanisms. And they published a second edition of it. This book was really well received by his friends and botanists. And I think that that's a really good evidence of the work that Darwin put into it. Here's a quote from Joseph Hooker, who's a really famous botanist at the time. The present work is one of a class of which botany possesses singularly few examples. I think that that statement could really be, you can think of that as kind of how to think about this work. This was an example of someone taking a theory and trying to provide a whole bunch of really good, detailed examples supporting it. So Darwin's goal in this book was basically to extend the arguments from Origin of Species and to try to show in much greater detail how natural selection could have kind of yielded these elaborate evolutionarily derived structures. And so one of the cool things about learning about Charles Darwin is that because so many have been interested in him for so long, all of his writing, for instance, has been digitized, and so we can access like his letters and stuff. And so here's a letter he was writing to Asa Gray, who was a botanist at Harvard University and another really prominent botanist at the time. He said, of all the carpenters for knocking the right nail on the head, you are the very best. No one else has perceived that my chief interest in my orchid book has been that it was a flank movement on the enemy. What Darwin is saying here to Asa Gray is, I put this book out as sort of a set of examples that show great or strong arguments, I guess, that my theory of evolution by natural selection, has abundant evidence to support it. Within this book, Darwin explored all kinds of orchids, both from Britain and all over the world, more or less any that he could get his hands on. And it included all these really detailed drawings. But he showed all kinds of aspects of the anatomy of these flowers and how it was related directly to pollination. So as an example of this, he showed that the flowers of the orchid genus Catasetum have this elaborate mechanism to actually launch their pollinia, which are these sacs full of pollen grains at bees. And these pollinia are really cool because they actually have an epoxy-like chemistry on this pad, so that when the polinium is launched from the orchid flower, it mixes two chemicals that quickly form a powerful glue-like substance. And the polinium attaches itself to the foreheads or the backs of bees. Darwin also showed for the first time that orchids had evolved the separation of sexes. So there are orchid species for which there are male individuals and female individuals, and they even have really different looking flowers. It would be quite easy to think that this Catasetum osculatum is actually two totally different species. Darwin was really fascinated by this because it allowed him to think about vestigial elements in these flowers. So elements are aspects of the anatomy that might not necessarily be currently serving, an obvious function. And so he said, at a period not far distant, naturalists will hear with surprise, perhaps with derision, that grave and learned men formerly maintained that such useless organs were not remnants retained by the principle of inheritance at corresponding periods of early growth. That's what his argument is, but were specially created and arranged in their proper places like dishes on a table by an omnipotent hand to complete the scheme of nature. What Darwin was referring to here is this idea that was forwarded by people that opposed evolutionary theory and evolution by natural selection. They argued that some of these strange elements that seem to be connected to evolutionary relationships are actually just sort of put there by a creator to trick us or to confuse us. One fascinating thing about orchid flowers is that many of them, though they attract pollinators, they don't actually produce nectar. Some of them even produce nectar spurs that don't have nectar in them. And in his earlier work, Sprengel had argued that these are sham nectar producers, that these flowers were tricking the pollinators. This was an especially challenging factor or facet of orchid flowers to Darwin, because it really contradicted his theory. You know, if these flowers were exquisitely adapted to attract specific pollinators to facilitate outcrossing, why would they be sham nectar producers? And, you know, so Darwin wrote, "He who believes in this doctrine must rank very low the instinctive knowledge of many kinds of moths." Darwin was saying, why would they keep visiting if they produced sham nectar? Here's a picture of a moth with 7 pairs of orchid pollinia attached to its proboscis. That would make life annoying. Darwin looked really hard to try to find nectar in these flowers, and he could never find it. But yet he was so convinced of natural selection that he just couldn't believe it was true. And eventually he came to argue in his book that the moths were piercing a membrane associated with the nectary, which unlocked nectar from a secret chamber. And you know, he wrote, I'm led to suspect that they penetrate the lax inner membrane. I'm aware that this is a bold hypothesis, for no cases are recorded of nectar being contained between 2 membranes, or of Lepidoptera penetrating with their delicate proboscis. So he kind of knew, like, he was really going out on a limb, making this argument. And it turned out he was just wrong, you know. He went to great efforts to suggest this, and this orchid clearly has a lot of contrivances to ensure cross-pollination. But it's true, it doesn't produce nectar, and the orchids are tricking their pollinators to some extent. Though there were some mistakes or errors in this book, really it's marked by triumph and incredible predictions. One of the most famous is his prediction about the pollinator of Angraecum sesquipedale. This is something that had actually been discussed. This orchid is really beautiful, and collectors in Europe were aware of it, and they were kind of trading it around. This is this orchid that's fairly common in Madagascar. And what's amazing about it is it has this really long nectar spur. So in the image on the right, the sort of green lines that are hanging down from below these flowers are the nectar spurs. That's a hollow tube that has nectar way down at the base. Darwin wrote, "I fear that the reader will be worried, but I must say a few words on it. Good heavens, what insect can suck it?" Well, what he was looking at that, and he predicted that it was pollinated by an undiscovered moth that had an 11-inch proboscis. You know, they knew of some moths that had like 6-inch proboscis, but nothing like that. And his argument was basically that there was an evolutionary race between these orchids and their moth pollinators. And what he was saying is that this orchid, by evolving a longer and longer proboscis, was basically encouraging more and more pollinators to try to reach that nectar. And so even if they couldn't reach it, they would still go in there and try to reach it, and in the process, perhaps pollinating the orchid. And it turns out that, you know, everyone was talking about this. And a couple of years after that, Alfred Russel Wallace, who I mentioned earlier, he predicted that it would probably be that this African sphinx moth, you know, a relative of this one that called Xanthopan morganii. And much later that it was found. And so it was named Xanthopan morganii predicta in honor of Darwin's prediction. And it turns out it took until 1992 to actually see this moth interacting with these orchids. But then it was noted. There they were with their 11-inch proboscis. The real strength of this book and of a lot of Darwin's work was just this really close observation and great amount of effort that he brought to bear. He spent a lot of time looking at orchids, dissecting orchids, taking notes, paying attention. So an example of this is, he spent a lot of time looking at these pollinia, these pollen structures of orchid flowers, and he was trying to kind of understand how they were working. And so he was always trying to simulate insects going in and pollinating. So he'd often use a pencil to do that. Here's an illustration of a pencil tip. And he's got a pollinium that has attached itself to that pencil tip. One of the things he realized only after a while was that if he looked at that pollinium after a few minutes, it had moved. Well, what he found was that the pollinium would attach themselves to whatever he was using to simulate a bee. And then after a couple of minutes, it would swing down. And after it swings down, it's suddenly in the position where it can connect or contact with the stigma surface of the orchid. So if this bee, gets a pollinium on it and it's still at the same flower, it's not going to self-pollinate that flower, because it's probably not going to be there for more than a couple minutes. But as it goes to the next flower and that pollinium comes down, then it's much more likely to pollinate. So all this close looking, guided by his theory, led him to make all these incredible deductions. Of course, some of them, maybe overly guided by his theory, were incorrect. Arguably, this work of Darwin's was the beginning of the biological study of coevolution. Coevolution is the idea that ecological interactions between different groups of organisms have some influence on the evolutionary trajectory of those groups. A classic example of this is pollinators and plants. And there's some examples of coevolution being quite close. This is an example of I guess now a decade-old study on coevolution between orchids and their bee pollinators. A long-held theory about orchid pollination, or hypothesis of orchid pollination, was that orchids and their bee pollinators might be pretty closely co-evolved. However, now that we're able to amass lots of phylogenetic evidence and use fossils and other mechanisms to date our phylogenies, so we have a time frame, what we can see is that the relationships between orchids here on the right and orchids on the left and their bee pollinators on the right, what you see is that there's this so mixed kind of jumble of connections here. And what that suggests is that basically there's been a lot of exchange or interchange in what orchids are connecting with what bee pollinators. The other thing you can see is that the bees have speciated or speciated more earlier than the orchids did. So you see the latest events of cladogenesis, of breaking into new groups on the bee phylogeny, somewhere around 5 million years ago. Whereas if you look at the orchid phylogeny, cladogenesis has continued right up until basically the present. And so this suggests that the orchids are evolving they're diversifying more now than the bees are, and they're kind of exploiting the preferences of those bees to get themselves pollinated. Oops. Another example of like kind of some more contemporary work on pollinators and flowers is this cool paper that was published a couple of years ago about Enothera, evening primrose. What they found is that the flowers of Enothera are able to respond to the sound of their pollinators and quite rapidly increase the concentration of sugar in their nectar. So these flowers basically, when they hear the vibration of a bee coming in, they can pump a whole bunch of sugar in that nectar so that they can serve it. Pretty cool. Our group at Stan has had some work, done some work on floral morphology with a connection to pollinators in mind. So the plants that we're working on are called the calico flowers. Calico flowers are in the genus Downingia. And Downingias are mostly endemic to California. There are about 13 or 14 species, and nearly all of them can be found in California, and only a few of them extend beyond the state. One of the cool things about Downingia is that they're mostly restricted to vernal pools. Vernal pools are the areas of moisture that develop during our winter in the rainy season, and then they slowly evaporate as the summer goes on. And as that evaporation process occurs, and as the temperatures go up, lots and lots of annual plants are able to germinate and go through their entire life cycles in just a few weeks. And that's the case for Downingia. In the picture below, you can see a couple of students, former students of mine, Antonio and Jessica, and the kind of pale purple blue around them is all Downingia flowers. That's at Kesterson, the Kesterson section of San Luis National Wildlife Reserve. We've been trying to characterize Downingia flower shape because we think that the flower shape should correlate or correspond with the pollinators. And we've developed this morphometric approach where we basically apply, we use images and we use these landmarks on the images to mathematically characterize flower shape. And one of the neat things about Downingia is that in many vernal pools, you'll see multiple species co-occurring. And one of our hypotheses is that co-occurring species might more strongly diverge in flower morphology when they're co-occurring than when they're separate from each other. The idea is that when the two species are co-occurring, there might be some kind of pressure basically to isolate themselves via their pollinators. And so our working hypothesis is that flower morphology in these two co-occurring species is more divergent in populations where they co-occur and more overlapping in separate populations. Sadly, because of the ongoing COVID pandemic, we haven't been able to do nearly as much field work on Downingia in the last couple of years. I really hope that we can get out into the vernal pools this spring and more in upcoming years. What we have been doing during the pandemic is to digitize our herbarium. So we were on a grant from the National Science Foundation recently, and what we're doing is we're part of a whole bunch of herbaria around the state, and we're shipping around this imaging center. So this is basically just a camera on a light box, but it allows us to take our specimens and make really high quality images of them. So there's Harmon and Justin there. They went through 6,500 specimens and took a picture of every single one. And it generates these really high quality images. And what we're doing now is moving those images into our web hosting platform so that when you search through the Consortium of California Herbaria there on the bottom left, you can see all of our records, and soon enough you'll be able to even see the actual specimens themselves without having to visit our herbarium. Hopefully, though, we can have you come visit in the near future if you would like. I think that, reflecting back on Charles Darwin and the work that he did in his book on orchids, and also trying to connect that and the themes of some of those themes of his life to what we do, I think what we've seen is that the level of detail that we can bring to the study of pollinator ecology and coevolutionary biology, is so much greater now than the tools we have are incredible compared to what Charles Darwin had in his time, and so I think we're kind of inspired by that close look. The other thing I want to emphasize is that Darwin was able to see all these flowers and learn about all these plants because of the connections, the professional connections that he developed. He had friends that he corresponded with, and they sent him plants. I think that's one of the other things that we have now that he obviously didn't have. These days I can email a big group of people. I don't have to write a letter and wait for that letter to arrive and then wait for a response several weeks later or whatever. So the possibilities in science are so much greater now. And hopefully we're really lowering the barriers to entry and making it so that anyone who's excited about it has a real opportunity to jump in and engage. And so I guess I hope that we can embrace Darwin's legacies there and help to, I guess, reduce the other legacies of the Victorian era that may still exist today in some cases. Thanks all. I'm gonna, I guess, say that I'm here at the end of my presentation. I think this floral diagram is cute. It looks like it's smiling at us.
That's great.
Here's some references, and I'd love to take questions.
Well, okay, so teacher me, we do plant collections every semester, and everybody has to key their plants out, and it's torturous. And I actually have some resources. I have them go on Kew Gardens website and look at their digitized specimens. Some that are there actually from Wallace and also Darwin, which is pretty cool. But I love the idea that there's going to be a local California consortium database. Do you know when that's going to be available online?
That's available right now. So if you go to cch2.org,
okay.
You'll find the portal there. And right now that's searching through, I think, 27 or 29 institutions worth of data.
Okay.
And all of our specimens are up there right now. So you can see stuff that Stan students have collected through the years. We just haven't gotten the images up yet. So that's the sort of most recent version of that project. And that's just going to be ongoing. But a lot of the institutions already have their specimens imaged and hosted up there. So yeah, it's a really awesome tool.
Yeah, a few years ago, I got to go and watch them scanning part of the collection at Edinburgh Botanics, and it was amazing. I couldn't believe it. It was just, but they had a lot of cameras. They were going, it might have been a little bit overkill, but the images after were it was almost like having the pressed plant right in front of you to look at. It was pretty great.
Like this lupine here, it's just one example, but this, the level of how far we can zoom in on these images and the kind of detail we can see is really incredible. It's
really fantastic. And people used to travel to go to these collections to do studies, to make cross-referencing, and they can go online now and lower their carbon footprint. They might every once in a while need to go see the real thing, but anyway. Okay, I have a few questions for you. One from Brennan, and he said, "Hi, Andrew. We talked about a couple of the hypotheses made were incorrect. Was there any significant backlash from making what Darwin considered bold claims, for example? How you mentioned his hypothesis that nectar may exist between a membrane layer in orchids?"
Yeah. So, Darwin was in constant communication with like all of the reviewers of this book and all of the people like out there that he wanted to get affirmation from, so
Yeah.
like all of us, and so it's funny, I haven't encountered anything from his correspondence that's like, sorry, my bad, about this. But
Right.
I think part of it is the sheer volume. When I first read this book, I was like, how is this a book for lay people? Like, this is just he called it a small book, but for Darwin, everything that wasn't like 2,000 pages was a small book.
Right.
And, the thing is full of these really detailed descriptions that are pretty dry. And
Victoria.
But I think that even though there are some of these instances where his arguments weren't correct, I think the overwhelming case is that most of them impressively were, and I think a lot of it is that, people form their ideas of things largely based on their preconceived notions. So, the going conversation in the world at that time, in the Western botanical world about orchids, was, basically that they were self-pollinating, they were just these like really cool, weird things there, but there's no way, we've never seen insects on them, so that was just the narrative. And so, to be able to change that narrative and turn it into something much more interesting, frankly.
Right.
and I think that people latched onto that and really did appreciate that. And I think that that's kind of how natural selection in general was received across a lot of the, we might say, progressive science biological community at the time. People were largely on board. And what it was about convincing some of the very vocal people that disagreed, and also kind of just starting to load it up with good examples of science to kind of follow up. And so, I think that largely it was successful. I mainly brought up this example because I think it's a nice example of how we are influenced by the theoretical framework we're working in. Darwin was like, they can't be just tricking them, like he became convinced, kind of almost overly convinced, that this was this really nice, close relationship. And so it just shows how we're all, heavily influenced by, the current state of affairs, and that even the best and sharpest among us are easily swayed into perhaps dangerous territory. And I think it's just a nice thing to remember. Thanks for your question.
Yeah, that was a good question. I love, for my botany people who are viewing, we will talk more about the pollinia when we get to evolution this semester. I always want to be able to share these great examples, even with my non-majors, like really to make, because they hear here's the evidence for evolution. And I'm like, you guys have no idea how big like this body of evidence is. Like there are these really cool examples and of adaptation. So yeah. And Denise is wondering, where are the vernal pools you're using for your calico flower research?
Yeah, so vernal pools exist all over California, but the ones that we're sort of focusing on for our group right now are vernal pools in the Central Valley, driven in large part by our ability to get there. But so we've been looking at pools at Kesterson, which is part of the San Luis National Wildlife Reserve.
Okay.
We've also been going to the Merced Vernal Pool Refuge. So the University of California Merced is adjacent to this big ranch that is now under control and is acting as a refuge. And that's just full of pools, and when you go to a lot of these spaces, what you realize is that there are thousands of these vernal pools. They're just all over the place in the springtime. And but if you get there too late, it's just the brown season and everything's just, all you see is like ruminant foot tracks, in the dried up mud.
Yeah, that's all that's left.
Yeah. But I'm working with, I'm kind of in talks with people at the San Joaquin National Wildlife Reserve right now, and I'm hoping that maybe we can find some pools out there that have Downingia. And we really hope to kind of expand the range, the geographic range of the species that we're looking at.
Excellent. Hey, if you guys ever have room on the bus for any of us MJC folks and students, we would join you, or maybe let us know. We could come along. That'd be fun.
That'd be really great.
Yeah, and then Denise just added that hopefully we'll get more rain this spring for the pools, and yeah, really hoping for some more rain. Well, I have to tell you, I am really thankful for you joining us today for and giving this Darwin Day talk, and that's probably us for the day. I want everybody to know I sat and ate all these during the there's none left for anybody. Anyway, thank you so much, Andrew, and I hope you have a great rest of your spring.
Thank you. Yeah, you all as well.
All right, everybody, this talk will still be here, so if you want to come back and check out this recording in the future, it'll be on the MJC Science Colloquium page. And if you would care to subscribe to our channel, we have a lot of great talks coming up in the future, and I hope you guys will join us again. Thank you, Andrew. Thank you, everybody. Have a great afternoon. Bye-bye.